In this section, we will talk about the diagnosis and treatment of the HIV virus. The diagnosis of HIV infection is usually established by indirect detection of specific serum antibodies to HIV by enzyme-linked immunosorbent assay or ELISA test. The test is performed in 3 steps. Let's understand this test. The test vessel is well coated with HIV antigen. Then patient serum is added, in which HIV antibodies are there. These HIV antibodies bind to HIV antigen in the coating of the test vessel. In second step, the bound antibodies are labeled using an enzyme-conjugated antihuman immunoglobulin. In third step, chromagic substrate is added to test vessel. The enzyme forms a colored reaction product in the presence of HIV antibodies. Then the photometric determination is done. A positive ELISA test should be confirmed by at least one other test, such as Western blot or a specific immunofluorescence test. Viral culture is a rather expensive technique and is not used for routine HIV diagnosis. Genes amplification techniques, such as polymerase chain reaction or PCR, and reverse transcriptase PCR, are currently being tested as HIV screening test. PCR is used to detect HIV proviral DNA, and reverse transcriptase PCR is used to detect viral RNA. Now let's discuss the treatment strategies of HIV. First is antiviral drugs. Let's think together which points can be blocked. First, blocking of catalytic center. For example, by nevirapine. Other possible mechanism could be incorporation of wrong nucleoside leads to strand breakage. For example, by azithromycin. Third is blocking of catalytic center because of similarity with cleavage site. For example, by saquinavir. These are all mechanisms for antiviral activity. So these are antiviral drugs. Second strategy is vaccine. The use of attenuated virus is too risky because the immunity observed in the animal model, such as monkey, cannot be extended to human. The development of hybrid viruses has opened new possibilities. The HIV genes for glycoprotein 120 can be transferred to harmless viruses, such as cowpox. This may permit the use of functionally weakened, but still highly immunogenic viruses as a live vaccine. So another approach is the use of purified DNA as vaccine. The injected DNA is incorporated in the cell genome. It can imitate the events of viral infection without virulence. Let's understand this. This is viral plasmid. It is incorporated in host genome. Translation occurs. Then viral-free proteins are produced that leads to humoral immune response. Now, endogenous processing in rough endoplasmic reticulum occurs. Then binding of antigen with MHC1 molecules. This leads to cellular response. Third strategy is through interleukins. When antigen binds TCR on CD8 cell, the activated CD8 T cell produce interleukin-16, or lymphocyte chemotactic factor, which binds infected CD4 T cell infected with HIV provirus, leads to signal transduction and decrease in viral production. This viral production has been decreased by 1/10. The chemokines, such as macrophage inflammatory proteins alpha and macrophage inflammatory proteins beta, have similar effects on T cells. Due to blockage of chemokines receptor required for HIV docking, recombinant interferon alpha and interleukin-12 inhibit number of steps in replication cycle, so they are currently being used in conjunction with reverse transcriptase inhibitors for treatment of asymptomatic HIV-infected subjects in experimental combination drug therapies. This was all about this section. In our next section, we will talk about the overall picture of HIV and AIDS.